Welcome to this lesson on RNA structure. RNA, or ribonucleic acid, is a single-stranded nucleic acid that plays crucial roles in cells. Unlike DNA, which stores genetic information in a stable double helix, RNA exists as a single strand that can fold and perform various functions. Understanding RNA structure is fundamental to grasping how cells express genes and produce proteins. The most fundamental difference between DNA and RNA lies in their overall structure. DNA forms a double-stranded helix, where two complementary strands twist around each other. In contrast, RNA consists of just a single strand of nucleotides. This single-stranded nature gives RNA unique properties and allows it to perform functions that DNA cannot. DNA and RNA differ in the sugar component of their nucleotides. DNA contains deoxyribose, which has the chemical formula C5H10O4. The prefix deoxy means it lacks one oxygen atom. RNA, on the other hand, contains ribose with the formula C5H10O5. This ribose sugar has an extra hydroxyl group, or OH group, attached to it. This small chemical difference has significant implications for the molecule's stability and function. Both DNA and RNA use 4 nitrogenous bases, but there's one key difference. DNA uses adenine paired with thymine and guanine paired with cytosine. RNA also uses adenine, guanine, and cytosine, but instead of thymine, RNA uses uracil. So in RNA, adenine pairs with uracil, while guanine still pairs with cytosine. This substitution of uracil for thymine is one of RNA's defining characteristics. You might wonder why RNA evolved to be single-stranded. The answer lies in functionality. RNA needs flexibility to perform its diverse roles in the cell. Being single-stranded allows RNA to fold back on itself, creating complex three-dimensional shapes. These shapes enable RNA to interact with other molecules, catalyze reactions, and carry out specific functions that a rigid double helix could not accomplish. There are 3 main types of RNA in cells, each with a specific role. Messenger RNA, or mRNA, carries genetic information from DNA to ribosomes. Transfer RNA, or tRNA, brings amino acids to the ribosome during protein synthesis. Ribosomal RNA, or rRNA, is a structural and catalytic component of ribosomes themselves. All 3 types work together in the process of making proteins. DNA is significantly more stable than RNA. The double helix structure of DNA protects the genetic information stored within it, making DNA ideal for long-term storage. RNA, however, is less stable due to 2 factors. Its single-stranded structure leaves it more exposed, and the extra hydroxyl group on the ribose sugar makes RNA more chemically reactive. This instability is actually advantageous, as RNA molecules are meant to be temporary, performing their functions and then being degraded. Let's recap the key differences between RNA and DNA. First, RNA has a single-stranded structure, unlike DNA's double helix. Second, RNA contains ribose sugar with an extra hydroxyl group, while DNA has deoxyribose. Third, RNA uses the base uracil where DNA uses thymine. And finally, RNA is less stable than DNA, which suits its role as a temporary functional molecule. These differences make RNA perfectly adapted for its diverse cellular functions.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.